Okay, thank you so much. A great opportunity given to me to share really an overview on Sanger sequencing, which is actually a gold standard in terms of detection and validation of SNPs, despite the emergence of next generation sequencing. So in the next 10 minutes, I'm going to bring you through a brief history about Sanger sequencing, how it works, the applications, and at the end, provide some opportunities CRID is actually offering to the scientific community through the AVecGen project. So it's really important to first understand what is DNA sequencing before understanding what is Sanger sequencing. So DNA sequencing is just a process of identifying the exact order of nucleotide encoding a specific genomic information. And talking about the method used is the Sanger sequencing that is actually used to identify the order of nucleotide basis in a DNA based on a technology which is the core, the heart of the method, which is called the chain termination method based on a modified nucleotide called dideoxy nucleotide triphosphate. So the method has been developed by Professor Frederick Sangers with his team when he was doing some experiment in the lab. He was actually able to introduce in a normal PCR reaction the dideoxynucleotide triphosphate that will actually block the extension or the polymerization, of the DNA, preventing the binding of the next DNA. So this, with this method, which is actually highly accurate, he succeeded to sequence the first ever genome of the 5X174. And that was this discovery actually marked the foundation of genomics as well as a genomic revolution. So there has been significant progress since the discovery of Sanger sequencing, leading to the sequencing of key organisms, including human mitochondrial genome, as well as the human genome in 13 years between 1990 and 2003. A task that can be actually done using a novel sequencer, next generation sequencers, such as the NovaSeq 6,000 in just 3 days. So about the principle, how it works. So it's just like in a normal PCR reaction with the common reagents, but adding on top of that a fluorescent label dideoxynucleotide, like I said, that will terminate the chain reaction and prevent the binding of the next nucleotide, generating various fragments of different lengths that are actually run on a capillary electrophoresis, and then at the end, there's a fluorescent detector that is used to identify the dideoxynucleotide triphosphate label, generating as well a chromatogram revealing the DNA sequence by reading the order of the fragments. So I invite you to listen this
foundation and gold standard
video.
of DNA sequencing since 1977. While new sequencing technologies have emerged since, Sanger is still an invaluable tool for all kinds of labs. The basic steps of Sanger sequencing are simple. The reaction involves template DNA, primer, DNA polymerase, nucleotides, and fluorescently labeled dideoxynucleotides. First, the DNA is denatured and then cooled to allow the primer to bind. The DNA polymerase enzyme then lengthens the strand according to the DNA template until it randomly places a dideoxynucleotide. The dideoxynucleotide is missing the hydroxyl group, which allows the next nucleotide to bind. So once it is placed, the chain is terminated and will not grow any longer. The color of the fluorescent dye on the dideoxynucleotide represents the last base which was added to that fragment. This reaction is repeated until the sample contains the fragments of all different lengths, each terminated by a fluorescently labeled dideoxynucleotide. When the fragments are separated by size, the dyes can be read in order to give the sequence of the template DNA.
So concerning the application, Sanger sequencing has broad application, including clinical application as well as research purposes. And some few example amongst them, if you are a human biologist, you can use that to detect key genetic variations and validate them from the next generation sequencing final output. You can use that for identification of parasite if you are an animal biologist. If you're a plant biologist, you can use that to identify plant species. If you're a microbiologist, you can use that for bacteria species identifications, as well as verification of plasmid construct. If you're a biochemist, you can use the SANG-Seq to sequence PCR products and also to genotype microsatellites. If you're an environmental biologist, you can use it for validation of organisms detected in environmental DNA studies. And tremendous strength of Sanger sequencing that are actually even surpassing the weaknesses. For example, it can generate very long reads and accurate outputs despite the low throughput it can actually generate after sequencing. But there's also key improvement, including automations of the technology to allow really processing of a huge amount of samples. So the global market of Sanger sequencing has been estimated at around 3.73 billion in 2025. And what is interesting here is that America alone is dominating the market at around 43.7%. And this is just because most of the company providing Sanger sequencing services are located in the North America. And what is really alerting here is that Africa is lagging far behind, and I think is the time to change actually the paradigm. And CRID is actually well placed to seize this opportunity because it's predicted that this growth will reach 12.17 billion by 2032. And we hope that all those eminent experts that are actually connected to this call will actually engage with CRID, collaborate, and converge their samples. Samples as well from the south should be converged at CRID and even from the north in a long term, so that can really contribute in supporting African and also globally researchers in making their genomic research. So Sanger sequencing is actually highly accurate compared to the NextSeq in terms of comparisons. It generates long reads compared to the NextSeq. And also the throughput is the key limitation. The throughput is very low when it comes to Sanger sequencing compared to the NextSeq, which can actually generate millions and millions of outputs. And the cost is actually higher, sorry, for the SANG-Seq compared to the next generation that can process really very large volume of samples, and the turnaround time is lower for Sanger sequencing, but higher for next generation sequencing. And the data analysis is actually straightforward with Sanger sequencing, whereas with next generation sequencing is actually complex and deserve key expertise in computational biology as well as in bioinformatics. So this is the last slide where I'm actually giving the opportunity that CRID is offering to the scientific community as well as our collaborators in terms of Sanger sequencing services, as our aim is actually to advance the local research in genomics and molecular biology, such a way researchers, scientists could make some really interesting and relevant discovery that could impact global health and also improve the well-being. So the first thing we are ensuring here at CRID is that we are eliminating the bottlenecks associated with sample shipping abroad, and we ensure affordability of the costs of sequencing here at CRID. My colleagues will be presenting on that after me. And we are minimizing the turnaround time to get the result once your samples are here at CRID. We are also providing very close and direct local support to all our collaborators. And also we are offering on-demand Sanger sequencing analysis, as well as capacity building of our collaborators, as well as scientists across the world. And importantly, we are interested as well in collaborative projects with public or private universities, hospitals, public health agencies, as well as sister institutions. So if you are working on animal genetics, plant genetics, human genetics, you are doing some works that desire the need of Sanger sequencing or even next generation sequencing in the future, you are welcome to this, our platform, and let's just engage and save life through quality research. So I would like to thank really our founder, the GATE Foundation, as well as our partners, and thank everyone who really took his time to join this first webinar on Sanger sequencing. Thank you very much for listening.
Next one is Dr. Estelle Mewamba, who will be presenting on the Sanger platform at CRID, the instrument that we are using to perform the Sanger and what that equipment can perform, to give you a broader description of the type of research questions you can address with the ABI 3500 SL. Estelle, if you are ready, over to you.
Okay, thank you, Prof, for giving me the floor, as well as the opportunity to contribute to this first and exciting webinar. So this afternoon, I will be taking you through my presentation entitled Sanger Sequencing Platform at CRID, the AB3500 XL. And my presentation is structured as follows. So first of all, we have a brief overview of the equipment, what it does, some key tips for successful sequencing using the AB3500 XL, some research application that could be used through this equipment, and future direction, capacity building, and a conclusion. So all of you can all see on your screen a device that is blue and white. That's what we call the AB3500 XL. It's a high throughput 24 capillary genetic analyzer that was designed in order to enable researchers to decode with confidence the nucleotides that are found in different DNA fragments from different sources. And this could be done using two approaches through this equipment, either using the approach of Sanger sequencing or fragment analysis. And you all can see that we have a laboratory scientist that is performing some DNA testing on this equipment at the Center for Research in Infectious Diseases. So this equipment is manufactured by Thermo Fisher, and it has some key components that are really useful when we perform sequencing. We have the capillary through which different DNA fragments migrate. We have the polymer that serves as matrix, mimicking the same function as agarose gel that we use in our laboratory, that will enable the fragments to be migrated. We also have the cathode buffer as well as the anode that will create a form of electric field for the migration of fragments. So what are some key features of this equipment? It's high accuracy, as it enables more than 99% of read accuracy. That means that if you have a fragment of like 100 base pair, you have the probability that 99% of this nucleotide were accurately base called. So it's also a high throughput device, as it can enable you to run as much as 24 samples in a single run, compared to previous devices that were developed that enable you to run like 8 samples at a single run. It also has the possibility of having RFID tracking that enable us to track consumable use in real time and assess the quality and streamline maintenance of the equipment. So how does it work? It uses the principle of capillary electrophoresis. First of all, consisting in sample injection throughout the capillary under a electrical field. The samples injected will be separated, and later on, the fluorescence that will be emitted by each of these labeled fragments will be detected at the level of a laser, and with an integrated software incorporated on the instrument, we have a streamlined and real-time base calling of the different signal being detected at the level of the camera. So how does Sanger sequencing works? What is the workflow? So first of all, you need to have your PCR product or plasmid, depending on what you are starting with. Depending if you are starting with PCR products, you need to clean up your PCR product using Exosap IT kit or either a purification kit. And once you have those purified products, you pass to the next step, that is cycle sequencing, in order to generate your fragment that will be fluorescently labeled at the end. And here the chemistry we use is the quantum dot terminator chemistry. We have several others. That's the one we are using. And after that, you need to perform a cleanup of your sequencing product in order to remove primers or remaining DNTP that would interfere with your sequencing. And once that is done, your samples are being loaded on the equipment where they will undergo the capillary electrophoresis, and fragment will be separated by their sizes under an electric field. And with the integrated software integrated in the equipment, your fragment will be, they will perform a base call, and at the end, you will have the entire length of your sequence that you submitted at the beginning. So in addition to Sanger sequencing, we can use the AB3500XL to perform fragment length analysis. This is quite useful for those performing studies on population genetics using microsatellites. And how does it start? You need, first of all, to extract your DNA, after which you do PCR amplification, this time using primers that have been fluorescently labeled using one of the following fluorochrome. It could be the FARM, the VIC, the NET, the PET, or the X, depending on you. And it really offers the possibility to multiplex as much as 6 samples in a single run. So after that, you need to perform some sort of denaturation, and after they will be injected into the in the capillary where they will undergo fragment selection, and at the end, you will be able to have the fragments you are analyzing with their real length and further continue your study. So here I will just take you through a simple demo of what happened when the apparatus is being launched. So first of all, here is what we call the capillary. As you are seeing, it's been inserted into the equipment. Once the capillary are well installed, you need to add your polymer that will serve as matrix for the migration of the fragments, and later on the cathode buffer and the anode buffer that will enable the migration of your fragments under an electric field. So once that is done, the machine will perform a form of calibration to make sure everything is in place. It's checking, do we have all the items required to launch the run? And now you can now put your sample. That is being added is the plate with your samples in different wells. So now coming to some key tips for successful sequencing, it's quite important to start with clean PCR product or plasma or plasmid, as it matters and can highly affect the quality of the sequencing release. So ideally, you need to have single specific amplicon as shown on this agar host gel, where you can see single bands. And always run your PCR product on electrophoresis to really confirm the size and the specificity of the primers being used. And after that, you need to perform PCR cleanup. You can do that using either an enzymatic method that is widely used using the Exosap IT. Either you can do a PCR purification using a PCR product purification kit. you have 1 year illustrated. In case you have several bands and that you really want to sequence a specific band of interest, it's required that you should perform a gel extraction, and you can do that using either the kit from Gadji. So what also matters when we perform Sanger sequencing is make sure you have accurate quantification, as overloading or underloading your samples can affect the rate and the quality. Ideally, we recommend you to use spectrophotometric apparatus. You can use the qubit, that is well recommended, but for some lab that does not have, you can use the nanodrop, but bear in mind that nanodrop usually overestimate your DNA concentration. And you also need to pay attention when designing your primers. You need to have high quality primers, following some guidelines when you design the primers, as the length between 18 and 24 base pair, the GC content between 40 and 60%, and make sure you really have a primer that heals you specific bands to a specific targeted region you are looking for. So as research application, this device, the AB3500XL, can be used to perform Sanger sequencing and fragment analysis, and this can be applied in several research fields, starting by entomology, where you can use this to identify different species. You need to detect some mutation, maybe linked to a particular phenotype, resistance, or maybe a disease, need confirmation, population genetics, or short-tandem read genotyping for parental analysis. So the field is quite broad, and all of us, we should feel concerned. You can also apply it in the field of medical veterinary, in microbiology, for those working on antimicrobial resistance to detect some key genes that could be involved in resistance, in environmental testing in order to detect new pathogens or any sort of pathogen that could be in the water. You can also use it in molecular parasitology and virology, maybe to detect arboviruses, plasmodium, or other type of parasite that you wish. And even in clinical biology for the molecular diagnostics of some specific disease affecting humans, mother and children, as well as in agriculture to detect maybe disease infecting some plants, or to discover some new variants of species that are being raised. So what are the future direction and capacity building opportunity using this equipment at CRID? So first of all, the plan through the AVecGen project will be to train scientists, first of all, indoors, in how to use the AB3500XL, and also perform training of scientists from Africa in order to capacitate them in using next generation sequencing technology or the Sanger Sequencing Platform in addressing accurately some research questions. And we also intend to expand to other pathogens, tuberculosis, HIV, drug resistance, bacterial, parasite, and all sorts of things you can think. So we intend also to collaborate regionally to build a genomic surveillance network and foster international collaboration. So coming to the end of my presentation, we can say that the AB3500XL is indeed a powerful, accurate, and flexible Sanger sequencing and fragment analysis platform, ideal both for researchers and clinical innovation. It's a real state of art, and it will offer to the global research community the possibility to perform this Sanger sequencing and fragment analysis, starting from Cameroon and broadly in Central Africa and more widely again in Africa. It really combines accuracy and speed as well as flexibility, and the platform is open to collaboration and expanding the use of this device. So we would like to thank our main founder, the Gate Foundation, as well as all our partners that are being involved in this project. So on this note, thanks for your kind attention.
Moving to Murielle Wondji, and if she's ready, she can upload her presentation. The presentation is ready for Murielle. Over to you, Murielle.
Good afternoon, Prof. Thank you for giving me the floor. Good afternoon, everyone. Thank you to the scientific community for joining this webinar, first webinar at CRID. We move straight towards the sample preparation and the submissions which are key elements for us to be able to carry out that Sanger sequencing at CRID. My presentation will follow this plan. We browse the service that we are offering now at CRID and how can you request the service, move to the sample preparation and submissions, then how do you get your results and the different timeline as well. The service we are offering now at CRID is the Sanger sequencing. You may have seen the flyers around all the different forums. We want to sequence using the Sanger method, the purified PCR products or the plasmid. We are also offering purification methods and also the QC, but this will be at extra cost. Concerning the purification method, we do have the column kits or the enzymatic that is available. For the QC, we can use the nanodrop, the qubits, or the tape station, depending on the products that you want to sequence. The size as well matters. So contacting the service at CRID, this is the flyers I was talking about. The first option is the email address sequencing@crid/cam.net. And also we will share with you the QR code. When you scan, then it goes straight towards this platform of the Sanger sequencing. So after you have contacted the service, what we want to do with you is to make sure that we get together to agree on the service that you're really after. So after you have prepared your DNA, meaning extracting it and amplifying your target, then you want to purify your product. You want to clean up that product, which could be a PCR or a plasmid, by running your gel electrophoresis. So your gel should be run after the PCR and after the purification steps as well, so that you make sure that you really have a clean one band on your gel, as you can see here. The common say is garbage in, garbage out. So you really want to have this product right to request for the Sanger sequencing. Thus, what you want to do is to make sure from the beginning, when you extract your DNA, to have a good concentration, a good purified ratio, same as an efficient primer design, for you to amplify the right target. So it has to be specific, and the GC content has to be between 40 and 60%. Also, when you have run your gel, make sure that they are really appearing only the one band. You might have like smears or double band. If you have double band, you can go for the extraction using the gel extraction kits before you send your product to the lab. So concerning still the sample preparation, before shipping your samples, there are pre-lab steps that we really have to get clear. After confirmation of the requested service, meaning when you contact us, we have time to discuss on exactly what you would like CRID to offer to you. Then we will agree and issue an invoice that you will have to validate to allow us to go ahead. Before shipping the sample and even start conditioning the samples, you have to read the guidelines of the sample preparation and submission. These are available on our website and could be shared as well with you when you contact us about the service. So we touch about the sample requirement, the concentrations, the conditions, the labeling, and the shipment. Note that you really have to use a proper tight fit container when you want to ship your sample to us, meaning either a screw cap or 1.5 ml that we close with a click, meaning it's properly closed. Also, when your sample are packaged, make sure while shipping them, you have to respect the cold chain. You know, we are in Africa here, sometimes you can have extreme weathers like the heat that will destroy your DNA. Here on this table, you can see some guidelines about different concentration. For example, if you have a product with a concentration from 30 to 40 nanogram, this is per microliter. Then if you want to sequence for one primer, meaning either the forward or the reverse, you will have to send us 15 microliter of the purified product. If you want to sequence it both ways, that will be 30 microliter. If a product is around 100 to 200 nanogram per microliter, then this will be 25 microliter for each primers, meaning around 45 for both ways. You also have our contacts here on this slide, CRID, Kulu at the PO box. And if you need any more information, feel free really to get in touch. We are very open and quite reactive to give you the information needed. The sample submission starts by this Excel sheet that we will share with you for you to fill in some key information that we will need while running your samples on the ABI 3500 XL. We are advising to use no maximum than 5 alphanumeric character while, because we have to be able to identify those on your 1.5 ml. Also, you can use your initial, these are just suggestions, and the number. You are free to code your samples as you want, but at the lab level, we have to be able to differentiate sample 1 from sample 2 from sample 3, or primer 1 from primer 2. So this will be about the submission. Continuing the submission, when shipping the samples, make sure you respect the cold chain at all time. We really like the samples to arrive in the lab in good order so that we are able to work with a good samples. So for those in Yaoundé or even in Cameroon, you can travel to Yaoundé and drop off your samples at CRID. This will be handed to a designated staff. You can also send them through DHL or this travel agency, touristic voyage, anywhere in the country. And if you are out of the country, we advise DHL. CRID do have a DHL account that we could share with you if that would make it easier for you to ship the samples to us. But bear in mind, you are paying for the shipping cost. So the form, submission form, usually we share the link below with you for you to go on the online and fill in all the information needed. We do not want to receive samples at weekends or public holidays. So while communicating with us, we will plan for the shipment together to avoid this disagreement. Now, once the samples are at CRID, you can follow these different steps on this slide online. The minute we receive your samples, we notify you of the reception. Then an acknowledged email will be sent to your email box to let you know that we have indeed received the samples. Once we move to the lab processing, the system will be updated as well. And concerning the lab sequencing, particularly when we have received the purified product, then we move to the cycle sequencing, the purification, cleanup, and sequencing reaction. Then we go through the equipment now with the purified product for the proper sequencing, and the results will be sent to you. We aim to have a 7 working days turn around. These 7 working days will be counted from the moment we have acknowledged the reception of your samples in the lab. And the deliverable, the results will be presented as this different link. We will send you the link through your email, but you also be free to download the results on the website. Also, this form is just an example to let you know how this will appear when you open your different folder. This is just for the result for the Sanger sequencing, mainly sending you the ABI and FASTA files. But if you need help with analysis, my colleague Gadji has talked about the bioinformatics part of AVecGen. We are quite open to offer that service as well. I will finish here and thank you all for your kind attention. I will also want to thank our funder and all our partners. Thank you again, and we are waiting for the Q&A to make sure we answer all your queries. Thank you.
Thank you. Thank you so much, Murielle, for that very practical presentation on the step to be taken to prepare, first of all, the samples and to send it to us. And there were some questions you even addressed about the bioinformatics support we can provide to partners. And you said clearly we can provide that support. Of course, we have to discuss further and get into a collaboration, and this is well possible, and it was explained. So we are moving forward with the next and last presentation of the series, and it will be from Jeffrey Otto, who is actually in charge of the business development aspects of AVecGen, and you'll be presenting the customer journey through CRID Sequencing Platform. Of course, we talk about sending samples, but how can you make the payment to ensure that things are done properly without delay, and other elements about engaging with the companies that could help facilitate shipping. We now ask Jeffrey, if you can upload your slides and go through your presentation. Meantime, again, we encourage people, participants to present, to add a question in the Q&A. We already have some. While Jeffrey is uploading, yes, it's there. And there was for Abdi asking that he's an entomologist and is looking at a training that was said in French, but I'm translating, whether he could be trained to do the Sanger. As we say, we are open for collaboration, so you can contact the team and we can plan that, and we can tell you what to do. Jeffrey, your slide is up, but if you can put full screen, please. Full screen, and you take the floor. Over to you, Jeffrey.
Okay, thank you, Prof. And good afternoon to everyone, and thank you for this opportunity. Just to double check, is my mic okay?
Everything is fine. Really nice. Go okay. On, please.
Thank you. So today, through this webinar, I will be guiding you guys through the customer journey, through the CRID service. We talk about sample preparation. We talk about the broad aspect of the project, but now how can you afford, how can you request the services? So my presentation will go through the customer journey itself. I will browse again shipping and logistics, pricing details for the Sanger sequencing, and collaboration and partnerships, as some questions are coming up on the collaboration aspect. Now, how does the customer journey start when you're requesting our service at CRID? You just start with initiating an order that can be done via phone, and for international, for worldwide scientists, they can even send us an email with providing additional detail about the project they want to go through. Now, once we have received this order, we make sure that we have all the information and that we have all the details to initiate an initial quotation. But in case we do not have all the necessary information, we'll contact the customer again and to make sure that we fully grasp the scope of the project before moving forward. So once we have all the elements, we initiate an initial quotation for the customer to review. This is to give the customer an overall aspect of how much the project is going to cost, and if it's within his initial budget. If it's not, we can review and amend some aspect of the order to initiate final invoicing. So once the invoicing has been issued, the customer receives the invoice and he has to agree on the terms and conditions, the data policies, and pay within 5 working days. Once we have payment confirmation, the customer then sends his samples through this sample preparation guidelines previously outlined by Madame Wondji, and we at CRID, we make sure to receive them and to check if they are viable and process them. In case they are not viable, we contact once again the customer and with an initial report stating the reason why the samples are not viable to move forward. So once we have checked and verified with the customer that additional samples or more elaborate preparation methods have been applied, we receive the viable samples, and then we move to the sequencing and the result. So at this step, once we have processed the samples and initiate sequencing, the customers then receive an email with the downloadable link where it can get a report of the project and the raw data of the samples analysis. So that's broadly the customer journey through the Sanger sequencing services. Please bear in mind that throughout this process, communication is really key, because we exchange throughout the work sequence flow. Now, talking about shipping and logistics, we are based in Cameroon, which is in Central Africa, and we have developed partnerships with logistic partners, essentially DHL for international purposes, where you can get a quote on the online platform if you are not based in Cameroon or even worldwide. We also developed a local partnership with Touristic Logistic, which is based in the 10 regions of Cameroon, and you can even send your sample at as low as 3,000 francs CFA. It depends, it's up to you. You can also drop off your samples, but bear in mind that you have to drop those samples on working days, not on holidays and weekends. Now, regarding the pricing what's included on the previous presentation, we talked about the Sanger sequencing price, which is 9,000 francs, on average $15.99 US. So what's included in this price? We have in this price sample reception and pre-processing, cycle sequencing on one direction, either forward or reverse, it's up to you. Capillary electrophoresis with the ABI, base calling and quality score assessment, quality control, manual examination of each trace file, validation and uploading of result. So that's what is included in the 9,000 francs CFA, which is roughly $15.99 US. However, what's not included in this price are the following: DNA extraction, PCR product purification, PCR analysis, bioinformatic analysis, and shipment fit. Please bear in mind that although these are not included, these are services available at CRID, and we can perform them for an additional cost. So it's up to you. to let us know what is the scope of your project and what you are trying to achieve. Now, regarding discount and bulk orders, we do offer discount for large sample volumes, and bear in mind that our pricing strategy is fully transparent. So you will not pay for services or for some things that we did not agree upon. Now, collaboration and partnerships. As an innovative hub and the first one in Central Africa, collaboration is a key aspect to this project. So we want to collaborate with you, with universities, research institutions, and also we want to collaborate with even scientists and researchers who have a specific thematic project and who want to take advantage of this genomic hub, maybe to go through the project or even article publications. Please bear in mind that we at CRID, we have multiple researchers and we have published several articles on well-known journal reviews. So these were the key aspects of my presentation regarding the customer, the shipping and logistics and the collaborations. And I would be happy to go through more details through the Q&A sessions. And thank you for listening and thank you for the opportunity given.
Thank you, Jeffrey. And if you can stop sharing, and I would like to upload the original presentation. Yeah, we are waiting for the ability to share. It's still not possible here. Just to maybe thanks everyone again, and then we could move to the Q&A. And I wanted to originally share maybe Estelle's online. If Estelle can share the original main presentation, that will allow us to touch some of the initial point where the quality was not the best, but we think things are better now. Estelle, if you can upload the master file, that will allow us to touch some of the points to provide better clarity to our participants. Meantime, we have some questions that we can start addressing. If you allow us to move to that level, we have a question from Grégoire Sawadogo. I imagine that it's from Burkina Faso. I might be wrong, but that's my guess. The question was, thanks for the nice presentation. This is really a progress having sequencing facility. So in terms of collaboration, how do you manage for the shipping challenges? I think that was before the presentation from Murielle, and I think you got an answer when she presented the way you should, you should actually prepare your samples for a safe shipping in keeping the cold chain. So we will come back to the Q&A, but maybe the first previous, that slide, Estelle, it was just to provide to you again where we started, hoping this time that the quality is better. I was highlighting the power of genomics and what led us to be involved in this field. We are heavily working on infectious diseases. Among them, you have malaria. And malaria is prevented mainly by using insecticide-treated nets, but mosquitoes have developed a way to avoid being killed by those insecticides. And the answer is in the DNA. And that's why we all need to assess the DNA for most of our diseases if we want to have an understanding of the mechanism driving a phenotype. You cannot avoid looking into the DNA. And this is all about what we are talking about when we talk about genomics. So in our case at CRID, we were tackling that question of what are the genes that the mosquito are using to become resistant. We assess the genome, the full genome of the mosquitoes, comparing those that are resistant to those that are susceptible to insecticides. And we were able, using this approach, to detect large regions. But when you detect the regions where the genes might be located, it's only a first step. You still need to put your finger on the specific gene. And often to do that, you need to use the Sanger sequencing. And so you can see that the sequencing platform are complementary. The next generation sequencing and the Sanger sequencing all work together to allow you to get the final answer. So when we detected the region, we also have a list of potential candidate genes, which we move to Sanger sequencing to now sequence a bit more samples. Because with the next generation sequencing, you cannot sequence a lot of samples. You are a bit restricted because of the cost. But with the Sanger sequencing, you can do more. That's what we did. We selected the candidate gene that we call a cytochrome P450 genes, and we sequenced mosquitoes that survived when exposed to bednet to those that died. And we were able to detect a striking amino acid change. That was a nucleotide substitution that led to an amino acid change. And we link it to the phenotype. We designed a DNA-based assay, and that was really powerful to tell whether mosquitoes can survive or die when exposed to bed nets. And we cracked the spread of that mutation across the continent or across Cameroon. And this is really the power of genomics, combining the whole genome sequencing, the transcriptomic, because we also do RNA-seq-based transcriptomic. And we have been doing that for years now at CRID, since CRID was set up in 2018. But although we did the lab work, we always send the samples in Europe for sequencing. And sometimes you will sit on those samples for 6 months, you don't have your data. And we were a bit frustrated. And thankfully, the data we generated, the evidence we show that we know how to do the genomic work in the lab, the wet lab. We know also how to analyze the data when we receive them back from the sequencing facilities. So we have the molecular biology, the genomic. We also have the bioinformatics skills. As you can see, all these were analyzed at CRID by the teams here. You have Mahamat, but many others in the team. That's why we can confidently say that we can collaborate with the community to analyze the data once sequenced. And how can we fill the gap? We were able to convince the Gates Foundation, and we really want to thank the Gates Foundation for awarding this grant to CRID to set up this facility that can serve the entire community, not just in vector control, but in everything. I think my colleagues explained to you that once you have the DNA, it doesn't matter where it's coming from. I think that this is where we are. Next, please. I don't need to go into this. The next, please. Yes, this is the slide I wanted to show that we want to cover all this. We already acquired the NovaSeq, which is a powerful next generation sequencer from Illumina that will sequence the human genomes. You can imagine it's large genome, 3,000 megabases. We are starting the next generation sequencing very soon. There will be a webinar on how you can do your next generation sequencing at CRID through AVecGen. That was not the purpose for today. Today we were starting with the ABI Sanger sequencing, and I think you had a clear explanation. And we are doing the long read sequence, short read with NovaSeq, but the long read also will be possible, and the platform really will be there to show you to allow the community to address many research questions that you have, but you are not able to achieve because either you cannot sequence often or quickly. So that's the overall of the presentation that you may have missed because of the quality. We will stop here and go back to the Q&A, hoping that this time the quality was better to allow you to see what we want to do. So if there's no comment at the moment, we might open the floor to the community. Maybe if someone has a burning question, we can give him the mic. Is it possible, Estelle, someone could raise the hand and we give them the floor for a question Prof. Before we go back to the question in the chat? Yes, we have. I will start just by order. We have Grégoire, and then Adrien and Prof Felix, a good friend of mine. So I will start with Grégoire.
Okay, morning, Professor. Morning, everyone.
Morning,
Thank you very
Grégoire.
much for this nice presentation. I'm Grégoire Saodogo. I'm from Burkina Faso. So I was asking, like, in Africa, sometimes even for the extraction, the different method that we use for the extraction, like the Kiagen kit or other reagent, it might be really difficult sometimes to get them on time and then, how to say, prepare your sample and send them to CRID. So I don't know if at CRID we have this kind of facility where you can facilitate those reagents. I guess that you have a partner that can help you to get them quickly. And then also maybe if I'm in Burkina, by this way, I can get the reagent quickly and then prepare my sample and send them for sequencing. Yeah, this is one
Yes.
question that I want
Supply
to
chain. Supply chain is the big issue, Grégoire, and I fully understand your frustration. But it has to come through a collaboration because we have to pay for you to reimburse. So this is part of the things maybe we should discuss in maybe inbox and find out how we can do this. But we cannot set up a service like that for the community. We cannot purchase reagents for the entire community. That we don't have the budget for it. But if we have a collaboration in place on a project, we can have an agreement to see how some of the reagents could be purchased from CRID and you reimburse with the mechanism that we can agree upon. So that's what we can say. And that's the case for all the community. We can collaborate and the collaboration will tell how far we can go. Thank you,
right.
Grégoire.
Thank you very much.
You're welcome. Adrien, Nana. Adrien, over to you. You can unmute.
Okay. Can you hear me?
Yes, good.
Okay. Thank you, Prof, for this opportunity. I'm a PhD student, microbiologist. I have two questions. The first, I want to make sure that I've listened very well concerning the price and the offer, because they say it's around $15.99 US, I think.
Yeah.
Now I want to make sure if it's for just one primers or both primers, because I think it's just for one primers.
It is for one direction. One direction.
Yeah, one direction. Okay.
Yeah.
I wanted to make sure. And now for other services like the previous person
Speaker.
have mentioned. Yeah, speaker have mentioned. The purification and other services, is it possible? Because sometimes we can have some strain and we do the extraction, but now about the purification, is it possible to have an idea of the cost of these services? Then we make us to prepare and make sure we are on the track of ready and on the track of what CRID can offer to us.
Thank you. Adrien. I think I will ask Murielle to answer that question about the other services, whether we already have a fixed price for it, because they might change depending of other conditions. But I will ask Murielle, if it's not ready yet, maybe we can find a channel through which we will communicate with the community what to pay if they are looking to do the extraction, the purification with us. And I think we discussed it, Adrien. Murielle will answer, but I'll make compliment later. Over to you, Murielle, or anyone, Estelle, who else can address that question? Murielle, over to you.
Okay, thank you very much, Adrien. This is very interesting questions. As we mentioned, we offer those services as extra, and at the moment, they are between 2,500 and 5,000. Once you contact us, then we can give you the exact price of, because we have to bear in mind the kit we'll be using and also the equipment you want us to use for your samples. So that's a bit what I can say about for now.
So that could be $5, $5 to $6, something like that. We will be doing the conversion. We know that the community is not familiar to France CFA. It's the local currency in Cameroon, but on the website, on the AVecGen website, we will upload all this information in dollars so that you are aware.
Thank you. Please, is it possible to finish my question?
Please go on.
Yeah, we have a big issue. I don't know about other community, but as Cameroonian communities, particularly PhD students, sometimes we have this lack of facilities where we can perform our works, particularly in terms of molecular thesis.
Yeah.
So now my question in terms of collaboration, is it possible for a student that want to perform the molecular part of his work to come and collaborate with CRID in terms of sequencing or even the simple DNA identification, and like my own, I'm working on resistant genes. So is it possible for a student that want to do that part to come and collaborate with CRID and make sure we can have a possibility of making our analysis there?
Yes, again, as we discussed during the various presentations, we are open for collaboration, for a partnership. And I think for that, you should engage with us through the emails or come at CRID. I know you're in Cameroon. We are really willing to collaborate, but it has to also be reasonable. We don't want to offer the moon if we cannot deliver. So it's about keeping it balanced, reasonable, but as much as possible, we really want to help the community either through the service directly with the term we explained, or through collaboration. And for the technicality, specificity, speak to us in, let's say, inbox, and then we will be able to tell you how far we can go, and also what is realistic. So engage with us, and the community is free to engage with us, and we will tell you always with openness what we can do and where we can work together. And if it's something we cannot deliver, we'll also be honest to tell you that we can't do that yet, or not at this time, because that community, that trust between us is key to go forward together. So thanks again. You are welcome to write to us later on. Thanks. We move to the next. I think Prof. Felix, over to you. You might be on mute. Felix? If not, we go to Linda, hoping that Felix may come back later. Linda?
Yes, Prof. Thank you very much, Prof.
Thanks,
thank
Linda.
you to everyone. Thank you for this excellent presentation. So my question is more about the quality of samples. So
Yeah.
I would like to know if, for example, the customer has sample done that exactly meets the required condition of the expectation, or some
I'm not getting you well, Linda. Linda, sorry, I'm not getting you well. Your line is breaking up.
Sorry. So, have you heard me?
Better.
Hello?
It's better now.
Okay, sorry for the phone. So, my question is more about the quality of sample.
Yeah.
I would like to know if, for example, the customer has a sample that don't exactly meet the required condition, then as well as the expected concentration or not clear bounds. Will you continue with the process, or will you simply notify the person, and if they give their approval, you then proceed with the sequencing?
Okay, Estelle?
Yes, there, Prof. So thank you, Dr. Linda, for your question. That's a nice one. So at each step, we engage with the customer. So once we receive your samples, a preliminary QC is being done. If they do not meet the desired requirement, then you will be notified through email, and you will be the one to tell us if you are okay for us to go ahead or we should stop there.
Okay. So a communication, a live dialogue will always take place, and between the customer and the AVecGen team to ensure that we agree before moving to the next step. Robert?
Sorry, Prof, I think Prof Felix is back.
Okay. Prof Felix, good to see you. Over to you.
Yes, sir. Yes, sir. Yes, sir. It's a pleasure to have you all. Thank you so much for this long run opportunity, because we have been looking for this for so long.
Thank you.
I'm very grateful to have this. Yes, I'm very grateful because some of my students and collaborators, even out of here, are connected. So I think most of the things I just wanted to share is written in the Q&A. And most of it is about how to get this collaborative work get started as soon as possible. As someone has already said, I'm very keen and eager to get collaborative work started, as most of my lab work, as I explained earlier, is on native and genetic resources from the land. So one of the key issues you didn't raise is about what we can call patenting, and that is one key. You may not address everything today, but maybe it's good
think about that.
to let us know. Yes, to know in the next presentation how to keep, because one of the concern why because these things, my personal PhD work happened on these 16 years back, but I think it was a dream to have it here done in Yaoundé, where we grew up each other. So that is something very wonderful. So congratulations to you and your team for that. That is a breakthrough. So so I have my queries are mostly two. Beside the patenting area, there is something key, is a kind of a training and capacity building network, because in our own context, in the university context, we have a high throughput human resources. So we are training one student, at the end of year is gone, and we have to restart again. So how can CRID be a part of a training? It could be online. We do not know. We don't have a lot of budget, but we can also hire. It could be part of tourism for you, maybe to come along and discover part of the country. So, or maybe student can also come that side. That is one. The second aspect is we have, let's say, the very key, the very prime steps before sequencing. So our lab is mostly dealing with this kind of stuff, DNA extraction and this, that. And thanks to Murielle, because of the presentation, we might be frightened and afraid that a lot of money can be put, and finally, CRID will say nothing is happening from from the sheet you sent. Excuse me, but just to how to make sure that maybe a kind of protocol could be shared. I mean, for us to be prepared on that, instead of shipping things to South Africa and everything. So if there are some documents, and if even on spot, because that we don't have a big bunch of money, but we have our small lab, and maybe we can have a kind of visit, and I can. So one. The second.
We take note of that. Yes.
Yeah. Mostly, they are not. This is what I said. They are not questions to be addressed right now, but you can think of it. And lastly is about to also deal with the project writings, because as I think a colleague just raised about the money, I think it was a PhD student. I think he's going through the same worries we went through maybe 20 years back, to get money to have things happen. So, but now that we have equipment, can we think of a kind of projects addressing specific, research project, and then where we can
work.
Part
of the collaboration, isn't it? Our partnership, and I think as we discussed, we are very much open to first of all generate the preliminary data with the community, and when needed to support the development of any proposal, providing our technical know-how, and then you probably will be the best in terms of the research question itself, but we can provide the support in developing the genomic aspects such that the proposal could be as robust as possible. I think we are willing to do that, but the partnership will really dictate where we go and how we do it. But we are open for collaboration, please. And even the training and everything, it will be the same as for AVecGen. We are willing to play that role. But again, I want us to be realistic, because we don't want to promise the moon and we don't give it. It could be frustrating. We will always do in the limit of our ability, but we are willing to really collaborate with you guys and engage with us online by email, inbox, and we will be able to tell you, even on the shipping of the samples, the protocols will be shared on our website, such that you know the protocol to prepare to extract the best DNA possible, and even the DNA purification, quantification, all the different steps. We have relevant protocols shared in our website, so that you can use it to maximize the chances to have the best quality possible. So this will be done, and we take note of that, Prof. Felix, having those protocols online to guide the community. Thanks for that.
Yeah, and we need it, please, very last one. We need it very straightforward, because as you know,
yes,
I have 38 samples of crocodiles.
Yes.
And so they have been sleeping a lot in my lab, and now it's time to get them out.
Absolutely.
So yeah, as soon as possible, if you can confirm when can we get started, and then I will order, because I didn't order some regions and primers, because I didn't want to have issues with electricity and things like that.
Of
And
course.
very last one, and that one is very urgent, because I'm applying for a short project for phenotyping.
Yeah.
And what you just presented, mostly the next point, and these short equipment, I think is very interesting, because in my small grant proposal, I would like to show that most of things that were supposed to be done in Vienna could be done here in Cameroon. So
exactly,
if you can agree and just send me what you have, and then I can quote and put maybe the website and I don't know your contact.
Absolutely. And we can confirm that we can do now the full suite of next generation sequencing and Sanger sequencing. Maybe you send us the research question you have, the type of topic you would like to address, and we can confirm to you which approach can allow you to tackle that question. Thanks.
Yeah, because
again,
I have only 2 days. Yes, to that. Thank you so much.
You're more than welcome. Welcome. And Robert, before we close with Prof. Ndo, is Robert around?
Yes, if not, allow his mic.
Okay. If not, Prof. Ndo, over to you. If you are any...
I've allowed Robert's mic.
Yeah, Robert.
Speak.
While we wait for Robert to take the floor, we have some other questions in the chat. One from Gael Chatue. If the shipping of samples takes more than 1 month, for example, does it affect the quality of the result? I think, Murielle, 1 month, what will you say?
Thank you for the very interesting questions. I know we are in Africa and the logistic is not always easy. We have faced that before, sending the samples to Germany from Cameroon. It took like 6 weeks and the results were not good at all. So I would say 1 month is a lot. We have to really make sure we can get the best possible channel, so that the samples will not stay more than 2 weeks in transit.
The shorter, the best.
The shorter, the best. Thank you.
Thank you very much. Next, still from Gael. I want to sequence the PCR products of a phytopathogenic fungi. How much money does it cost per samples? I think we answered that question already. $16 for per sample for sequencing. Is it possible for me to assist the sequencing of my samples? We, that will already be part of the collaboration. So if you want to be part of the process, that needs a collaboration in place with the CRID to do that. But the technical service itself is just to drop the samples and the team will work. We also have questions from Paul Alvin. Is it possible for us to directly send the collected and stored samples, frozen or ethanol preserved? Alternatively, will you prefer us to handle the purification? Maybe Estelle, could you answer that question from Paul Alvin?
Yes, Prof. So as we said previously, there's a possibility for you to be charged on additional services. But if you think you can do the DNA extraction, that's fine. But if you don't do it, then at our level, we will provide with you an invoice depending on the DNA extraction method and the additional services you are requiring, maybe PCR purification for you to pay. So that's an answer.
Thanks, Estelle. I think that I guess, please contact us. The emails are there. We can chat with you. We are open for that engagement. Still from another colleagues from Burkina, Aroua Traoré. I'm a PhD student in Burkina Faso. What are the conditions for collaboration? I will say no condition. Just send us an email, and from there we take it and see what we can do. I think that's the key. I will also like to know if it's possible to come for training. Yes, training. Someone also asked training and even data analysis. Mahamat, what will you say for data analysis?
Yes, Prof. Kant, we are really open to support our collaborators in terms of capacity building. We can, but it's actually on demand, so they can reach out to us and express what they really need to do, and we can organize that and see how we can get them trained in terms of data analysis.
Thank you. And
also to add, sorry, Prof.
Go on.
Yes, to add, we also have, as part of the project, we have, and I think we'll be launching from next year, we have some capacity building trainings that the call will be launched, and people will be able to apply and they will be shortlisted and trained in terms of bioinformatics aspects.
Exactly. Yeah. So we have a question for Prof. Ndo. Is there any process in place to avoid mix-up of samples on you when you receive it? Yes, Estelle, you receive the samples. Are you not at risk of mixing up and then sequencing the wrong samples for the wrong customer?
Not at all, Prof. So once your sample are being received in the lab, we make sure the matches what you have been sent, and the samples of each submitter is being stored separately. That means we process samples at once and make sure with our trained lab scientists, they make sure to respect the barcodes. So, and we have a design plate of each one that is being done. So the risk of mixing up sample is quite low, negligible, and
negligible.
Exist.
Exactly. Okay. Yeah. Thanks for that. Last question, maybe before we close or we get any final comment. We have Tessie asking she's based in Cameroon, in the northwest and southwest, where touristic a company might not be operating. Jeffrey, she's inquiring whether there's any alternative to touristic in northwest, southwest. Jeffrey?
Yes, Prof, thank you. Actually, with recent talk, actually touristic is based in Limbe and Boya, and they have an agency in Bamenda, but nevertheless, I have to engage with her to find out the specific times, because in those regions there are specific days for shipping out packages out of the towns.
I think Tessie got the message. And then the last one is from Kachuang first presentation talk about the partnership with researcher on animal genetics on Sanger sequencing. I'm interested. What are the conditions? I think just contact us inbox. We could establish a collaboration. I think if you are moving into the collaboration route, let's engage by sending us an initial email explaining the nature of the work you want to perform and what you would like to do together with us at CRID, and we will provide you the feedback and we take it from there. Thank you very much. I think we have answered most of the questions, and we would like to thank the community. At some point, we were more than 100, and we see this as the first engagement of many to come. We are glad to engage with you to provide you with the right service in terms of your Sanger sequencing to reduce your burden and the waiting time so that you don't have to wait for months to get your samples, your sequencing back. And we would like to thank the team, the speakers, Mahamat, Estelle, Murielle, Jeffrey, and myself as well.
Sorry,
yes?
Yeah, there are many people asking if we can share the presentation with them. I don't know.
No, we will. We actually recorded this and we'll be sharing on our website such that you have access to this. That will give you more detail, or maybe you can consult, you can look into this your free time, and that will also inform you what to do for your shipping, preparing your samples. So this will be made available to the community through our website. On that note, again, thank you so much. Let's together move genomics forward in Africa. First, starting with Sanger sequencing, but as I told you, just pay attention to the announcement. We'll be having another webinar in the coming weeks or months on the next generation sequencing facilities that we also have at CRID. And we'll be glad collaborating, working with you. All the best, and see you soon. Bye-bye.